Hello, I'm Smita Krishnaswamy. Today I'll be talking about some of the newer work in my lab having to do with deep representation learning for problems in biology. My lab tends to focus on machine learning and unsupervised learning techniques for big biomedical data. By big biomedical data, I actually mean any kind of data where there are a lot of observations and usually a lot of features. So one common data type we use in our lab is single-cell RNA sequencing data, where the observations are cells and the features are the counts of the mRNA molecules. We've also used other types of data like patient data, fMRI data, ETAC-seq data, and also data from structural biology, so graphs representing proteins and so forth. The interesting thing about this kind of biomedical data is it's completely open for exploration. And one of the main tasks that we want to achieve on this data is to understand the structure of this data. As you guys probably all know, biological data has a high degree of complexity, multi-levels of organization, starting from atoms to cells to molecules to tissue. And we want to be able to understand this kind of hierarchical structure, even on a smaller scale, in the systems we're studying in order to understand what the emergent levels of complexity are, how the cells are organized in tissue, for example, or just in order to explore the various biological entities that you're measuring. But some of the challenges in this kind of data include the fact that the data is very noisy. There can be a lot of it, so it could be hard to scale computationally. The high dimensionality can make the structure difficult to understand. So the goals of our research are to denoise the data and then to organize the data at multiple levels of resolution, and then to distill that organization into lower dimensions so that we can visualize the structure, and all the while be able to handle the increasing amounts of data that we get all the time, so millions of data points even. The outline of the talk is as follows. I'll talk about a unifying principle we use to achieve most of our goals, which is called manifold learning, and just some basics about it. Then I'll talk about eliminating noise with a newer technique called graph signal processing, and then organizing data into a multi-level hierarchy, and distilling the structure using our hierarchical visualization, and finally creating neural networks that are able to learn similar kinds of representations because we're putting these manifold learning multi-scale priors that work so well in biology. So the manifold assumption is this idea that even though the data is measured in high dimensional ambient space, that the intrinsic structure is lower dimensional and it lies in a smoothly varying sort of sheet that's coiled into higher dimensions, and it can be many such sheets or patches. And the idea is that because the features are all correlated or informationally redundant with one another, that the degrees of freedom are very curtailed for viable biological entities. And if we learn the shape of this lower dimensional space where the data lives, or the data manifold, then we can address a lot of the challenges that we have. And so one of the basic techniques for learning the manifold is the diffusion map technique. Here, if you have data that's observations by features, for example, cells by genes, we calculate distances, we convert the distances into affinities using a Gaussian kernel or equivalent kernel. And then you can Markov normalize these affinities to get basically a random walk matrix, or equivalently a diffusion operator. It's well known that the eigenvector of such a diffusion operator is called a diffusion map. And here the diffusion maps encode the nonlinear dimensions that are intrinsic to the data. So the intrinsic coiled dimensions of that Swiss roll that I showed you. So this can be a powerful way of learning what the key axes of variability are, even though biological data is often nonlinear. Another way of learning the manifold is to use an autoencoder. It's been shown that autoencoder neural networks, which simply recreate their input at the output, you can have an informational bottleneck where they learn these same types of meaningful intrinsic dimensions. And in this talk, we'll be using both the methods that I just went over. So one of the first big papers to come out of our lab had to do with denoising the data using these manifold learning concepts. And this is a paper called Magic that came out in 2018 in Cell. The main idea in Magic is to restore data to the intrinsic manifold dimensions using something called a low-pass filter for the gene signals. And I'm going to explain that a little bit. So if you have a cell by genes matrix, and the high values here represented by warm colors, low values by the cool colors, what you actually get is a highly dropped out or very noisy version of this, such that you can't tell which two genes are correlated or have relationships with one another. So you can think of this as sort of a noise. If you thought of it in terms of images, you see that the image on the left could be the original image, but if you had a lot of noise sprinkled on your image, it would look more like the image in the middle. Now in image processing, people have often used what's called a low rank approximation, or recreating the data from just a few eigenvectors of the data matrix. So we can think about using the same kind of technique here, except for what's normally used, which are singular vectors and singular values, don't really work well when you have these kinds of coiled dimensions, because if you have noise along the nonlinearity of the intrinsic dimensions, then these linear methods won't take it off. So what we do instead is we use the nonlinear dimensionality reduction that I told you about in the form of diffusion maps, and we take a more subtle approach than just throwing out some of these diffusion components or something like that. So for this, we notice that the diffusion components are eigenvectors of the diffusion operator, and also that they encode frequency harmonics. So you have the low eigenvalued vectors encoding long-range trends. For example, one end of the grid graph to another, it's slowly changing, whereas the last few eigenvectors with small eigenvalues encode fast-moving trends that oscillate a lot. And you can see these well on these very regular graphs like line and grid graphs, but you can think they're doing the same thing in your data graph. So what essentially we have to do is we have to take whatever signal we're denoising, which is the value of the gene in every cell, we have to load it to these eigenvectors. And loading it to these eigenvectors is also called taking the graph Fourier transform. And once we do that, we have to cut off the high frequency eigenvectors. And this is the operation that I call low pass filtering. And this takes inspiration from audio or image signal processing. And so if you cut off the part of the signal that loads to those oscillating high frequency eigenvectors, you get a much smoother signal. And so here, just to take a step back a little bit, we have a cell-cell graph. The nodes are the cell. They're connected to each other based on their affinity or similarity that we computed, as I showed before. And the genes are the values that are on the cell that are treated as signals, and then these are low-pass filtered. So the way that's actually done is first you create a low-pass filter. We create a low-pass filter by actually just taking these eigenvalues and powering them. Because this is a Markov matrix and it has eigenvalues going from 1 to 0, when you power these eigenvalues, they become smaller. So you see the eigenvalues are there, and when you power them, they become smaller based on what the value of t is. And then you apply this to your original data. So this would be like taking the graph Fourier transform of this signal, filtering, and then reversing that operation to get a smoother signal back. In the vertex domain, you see that this operation has the effect of looking smoother and smoother on the graph. And when we do this, we are able to learn the gene-gene relationships again, which tells us that we're learning meaningful values or corrections of the gene value. So here you see two gene-gene relationships. Initially, with all this dropout, it's very hard to tell what this relationship looks like. And as you low pass filter with a higher and higher t, you restore these relationships. And we've validated in data, for example, that doesn't have this kind of dropout, that if we artificially add it in, that the magic method can recover the data from the dropout and also any other kinds of noise that are in there. So magic involved one level of low-pass filtering of the data, and this achieved the first goal I talked about, which is to remove variation associated with dropout noise. But what if we kept filtering at multiple different levels? And our hypothesis is that this would remove additional variation and reveal other levels of structure in the data. So we undertook another project called Diffusion Condensation, which was published in IEEE Big Data last year, where we did exactly this. So the idea is to allow natural grouping of data points to appear at different levels of granularity by repeating the low-pass filtering. So how do we repeat the low-pass filtering? Basically what we do is, in that last step when we low-pass filter the data, we get what we call condensed data with some of the variability removed. Now you take this new data and you start the process over again. You calculate distances, affinities, and filter it again. And if you do that, you're applying this filter we created over and over again in a deep tree-like fashion that you see on the right. And this can reveal structure at different levels of granularity. Here's an animation of this. As you continue to iterate this process, you see that these data points are merging with one another. And eventually you'll see that the three different structures that are there, this is a mixture of three Gaussians, will appear. And if you continue to wait even longer, all the data points would merge into one single point, and that would be the end of the process. So this reveals data at all levels of granularity, and there are no forced merges. The points naturally come together. And then you can see what the salient levels that you'd want to interpret or further analyze are. One interesting thing is that this process actually creates a continuous tree, a little bit like hierarchical clustering, but here the lengths of the branches have meaning. The amount of time for which these branches stay apart tells us something about how distinct they are, and this is shown on a retinal bipolar data set, which we're further analyzing now. Another system that we applied this to is looking at neural connectomics. So we took the connectivity data of the neurons in C. elegans worm, a young adult worm and an adult worm, and we conducted this diffusion process based on how closely connected two neurons were, as in how often they traveled together, and how many times they were thought to synapse from these imaging data sets. And what we discovered was different levels of organization of these neurons. In part E, you see that there's a mechanosensory circuit. So the known circuit is given there. It consists of mechanosensory neurons and interneurons. But we were able to identify additional components of these circuits only through the diffusion condensation process. And our collaborators together with us have been investigating this, and we have a paper in revision at Nature that's looking at the different modules and neuronal circuits that come out as a result of this. So the next goal we had was to distill this data into viewable structure. For this, our lab has a method called FATE. I'm not going to go too deeply into FATE, except for telling you that FATE is actually a metric preserving dimensionality reduction algorithm that keeps global structure together. So FATE also has diffusion maps inside its engine, but what it does is, unlike a diffusion map, which puts different trajectories in different dimensions, it collects all that information nicely into 2 dimensions. And in the FATE paper, we showed it on this embryoid body data set, that it shows the differentiation structure going from the red human embryonic stem cells out to these various lineages. And FATE's the only one that shows the branching structure very, very clearly. So now we have an extension of FATE that we're working on called hierarchical FATE that combines FATE with the diffusion condensation process. On the left, you see original FATE on T cells, B cells, myeloid cells, basically PBMCs. On the right, you see hierarchical FATE, and hierarchical FATE shows you this structure in a much more simplified format, and the sizes of the dots show the number of merged data points at the level that we're drawing this. And we're showing clusters at a higher level that's automatically generated by the diffusion condensation process. So the advantage of this is that you could also zoom in. You can look at this overall structure, which is very simple, and then you could zoom in, for example, on the T-cell cluster that's to the left. And when you zoom in, you see this additional structure, that there's naive cells, Tregs, NK cells, the red CD8s, and so on and so forth. So this can give you a way of interacting with your data. And of course, this is not limited to single cell data. We've also looked at next strain COVID-19 sequences. So on the left, you have original fate, and on the right, you have H-fate, which shows you the simplified structure. And whenever you zoom in, you can see additional structure and components. So on the right is the zoomed in version of the orange cluster here. And finally, this method is pretty highly scalable because of some tricks we use to compute the diffusion operator at every point. So while we've done a deep signal processing based approach for this, we were interested in seeing if we can put this directly into a neural network. The advantage of neural networks is that they're very flexible. You can just penalize them into learning with fewer or more constraints. And you can examine features very easily. In addition to that, you get a lot of the scalability for free without some of the diffusion tricks that we used. So for this, what we're working with is this setting where we have not just one graph or tree of data, but we have literally thousands and thousands. These can arise in chemistry or biology as molecular graphs. So these biomolecular graphs can have hundreds of nodes, and they each represent a molecule. So this is taking the scalability to the next level. And each of these graphs may possess fairly complex properties, like can this graph, which represents a protein, bind to a certain kind of antigen or a compound, what's its energy when it has this kind of fold, etc. So on the right here, you see RNA secondary structure, and you see that some of these structures form hairpins and others don't. So what this requires is for us to be able to process a large amount of these graphs or sometimes trees and represent them in a graph-wise way. For this, what we use is something called a scattering transform. Scattering transforms have been around for a while, but they were ported to graphs in two papers by Feng and Gama et al. Last year. And what scattering transforms are multi-level wavelet transforms. So they're similar to the tree that we showed with the repeated low-pass filtering, except for they have high-pass and low-pass filtering and band-pass filters. And so the way that these are created is you take the difference of two scales of these random walk matrices. So these are powered to two different T's, but they follow dyadic scales. So they're powered to P is the Markov matrix, 2 to the J minus 1 minus 2 to the J. And these are different scales of wavelets. And when you take signals on a graph and you take these wavelet transforms, you get a lot of coefficients. And you can summarize those coefficients with a certain kind of pooling of statistical moments. So what we do is we take this graph scattering transform features and we auto-encode them. As I said before, the autoencoder learns the manifold, and we can also train them to predict the meta property of the molecule. And we can even generate off of this. If we sample somewhere in the latent space of the manifold, we can invert the scattering to recreate a graph. And we've shown this in a recent preprint that we're also going to present at the graph representation learning workshop at ICML. Here's a toy graph on top, which shows a linear trajectory. We created it to be a linear trajectory. We randomly modified one graph sequentially, and you see that the GSAE shows the linear structure of it the best. And here on the bottom, we have a set of graphs that all represent one RNA sequence called seq3, but it's known that this RNA sequence has bistable structure with 2 energy minima. And you see that you can see that structure of 2 bistable structures with 2 energy minima the best using GSAE, which is able to organize these graphs both by their similarity and by the meta property, which was energy here. So I hope I've shown you that manifold learning can help you address a lot of these challenges. We, of course, have a lot of other neural networks in our lab if you're interested in checking out this flavor of work. Manifold aligning GANs, autoencoders that organize data by archetypes, neural networks that model dynamics based on manifold constraints, and also sort of an end-to-end analysis framework called SST that can automatically cluster data. And if you're interested in any of the software algorithms I talked about, you can check out our GitHub page, our website, and we always announce on Twitter when we put in newer software. And if you have any questions, I'd be happy to take them. Thank you.